So now let's move towards the biosynthesis of these very important molecules. Before we talk about the individual reactions of the biosynthetic pathway of the glycoproteins, let's see that to which part of the cells they are transported, they are then located over there, and how this transportation takes place. You know that the Golgi bodies play a very important role not only in the processing of the protein but also in the translocation of the protein, especially the proteins which are destined towards either lysosomes or the cell membrane or even the ECM. You can see this in this scheme. You all know about this, that during the translation process, as long as the protein is being formed, it is also undergoes certain modification in the rough endoplasmic reticulum. Once certain modification have occurred in the rough endoplasmic reticulum, then they move towards the Golgi bodies and further processing takes place there. So you know about the glycoproteins, that the glycoproteins are made up of protein plus the oligosaccharides, and these oligosaccharides are then from the vesicles of the the Golgi bodies. They are transported either towards the outside of the cell to become part of ECM or to become the part of the cell membrane, plasma membrane, or they are incorporated into the lysosome. So this is the root of the glycoproteins and how the Golgi bodies are involved in it. Let's now see general scheme of the reactions which are involved. First we will see the general scheme, and then we will see the reaction at individual level. You know that there are oligosaccharide molecules which consist of different sugar residues. They include the UDP glucose, UDP galactose. Why UDP is there? Now it shouldn't be a strange thing for you. Then there are also mannose, also not only the molecules which we have just said. Then there are GDP mannose also, then GDP L-fucose also. So not only that UDP act as the molecule which will be involved, which will make it high energy molecule, but also GDP molecules are also there. And you know, I have already mentioned you in detail, showed you in detail that how NANA is different from other molecules because it contains CMP as the nucleotide. It doesn't contain GDP or UDP. It's perhaps the only molecule which have CMP as the nucleotide, which makes it a high energy compound. So protein synthesis takes place in RER, and the proteins are during translation they are moving towards the RER, that is rough endoplasmic reticulum. They move towards the lumen of the rough endoplasmic reticulum. There initiates the process of glycosylation, that is the proteins are then incorporated with the carbohydrate moieties and being added to the proteins. The first of the residues are the N-acetyl galactosamine, and then this N-acetyl galactosamine of course has to be converted into UDP N-acetyl galactosamine. So this is attached, this residue is attached with the serine residue, threonine amino acid rather, as you have already known that serine and threonine residues make one class, and then there is another class of amino acids which are involved in it. Again, as I mentioned, that very major portion of the processing and post-translational modification in the protein takes place in the Golgi apparatus. The stepwise synthesis of the oligosaccharide takes place. It's not that the whole oligosaccharide molecules at once gets bound with it. This specific order, I mean that which residue will attach at which sequence, it doesn't depend upon a template, as you saw, for instance, in the case of DNA replication or RNA transcription, that there is a specific sequence of nucleotides which are added where DNA acts as a template, or when proteins are being formed during the process of translation, then the amino acids are added where mRNA acts as a template, and for an mRNA, DNA acts as a template, and the amino acids are attached accordingly because of the sequence of the codons. There's nothing like this in case of this post-translational modification of this type here. In when we are going to make the glycoproteins or other conjugated molecules, it depends upon what specific substances attach at one time, and on the basis of this, that the cellular machinery then decide to add it or not to add it. There is also one thing we have to keep in mind, that of course this process takes place without any template, but again, then there are certain factors which have to be taken into account, is the cellular environment and the time which is going to be taken by this process.